Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today I'm delighted and honored to have Dr. Hasmukh Jain, who is the Professor of Medical Oncology at the Tata Memorial Hospital. And he has been instrumental behind India's first homegrown gene therapy for cancer, developed by IIT Bombay, Tata Memorial Centre, and ImmunoAct, called Next CAR-19, which was launched by the President of India, Smt Droupadi Murmu, and it claims to be the world's most affordable CAR-T cell therapy. The topic of discussion today is going to be low-cost, indigenously built CAR-T cell therapy in India. So, Doctor, really appreciate you taking time being part of the podcast. It will be great if you can start by explaining the difference between cell therapy, stem cell therapy to gene therapy. How do they actually work? And what role do you see of these cell therapy, stem therapy, and gene therapy playing in the future of healthcare?
Right. So these are all different aspects, different components of the broad spectrum. The broad umbrella. They come under the category of cell and gene therapy. Now, simply cell therapy means that you're trying to modify the existing cells of the patient in different ways, so that they would either express the protein of interest, or you're doing some manipulation so that they become more effective in acting against the cancer cells, for example. So not necessarily this is only for cancer, but this primary success so far is seen in cancer. But then also, in principle, this can work in autoimmune disorders. This can also work in degenerative disorders. But the cell therapy is essentially you're modifying the cells, and then you're giving it back to the patient. Stem cell therapy is where you're trying to harness the capability of stem cells. Now, we know that in body there are some of the cells. So regeneration of the cells happen because there are stem cells. Stem cells are the ones which make sure that once any cell is dying off, you have this reservoir of cells in a body which will then regenerate and form new cells. It's like it's a reservoir, and it can differentiate along different cell lines. So it can either form your let's say skin cells, it can form your blood cells, it can form your cells in the eye, etc. So it has potential to differentiate into any of them. So in principle or in theory, what the stem cells are meant to do is that you're depending upon what disease you're trying to target or what cell you're trying to create, you're introducing these stem cells, and then you are trying to manipulate the stem cells so that they will form that particular cell subtype and help in the healing process. So that's an example of stem cell therapy. Most of it is under research, but this is something which is very attractive. Gene therapy is where you're manipulating the gene at the genetic level. You're manipulating. So, for example, let's say there is a defective gene which is causing a problem. So you're trying to replace this defective gene with a normal gene, or you're trying to introduce something which will reset this gene's function and will again become normal. So these are the ways these therapies differ. But in essence, they're all trying to manipulate the cell system, the genetic system, and trying to cure the diseases.
Doctor, really appreciate you kind of explaining the difference between cell therapy, stem cell, as well as gene therapy. This, I believe, will fundamentally completely upend how we do healthcare. So very excited about it. You explain cell therapy. Will be great if you can also explain CAR-T cell therapy. And will be great if you can talk about how the idea generated of building a CAR-T cell therapy and the challenges of developing a CAR-T cell therapy here in India.
So the first thing is CAR-T cell therapy essentially means it is chimeric antigen receptor T cell. So what it does is you take T cells. Now our immune system has different components. There are different cells which represent our immune system. Just like you can imagine the defense of a country. So there are different problems. There are certain problems which are local, like your cops will handle, the police will handle, and then there's certain specific which is at the level of the country. And again, within these defense mechanisms that are there in place, there are certain components of the defense which are non-specific. So any kind of whenever there is any kind of danger, they will just go all out and they'll go destroy them. Whereas there are certain components of the immune system which are very specific. So what they will do is they will go and specifically target a particular problem, and there will be very minimal collateral damage. So exactly in the same way, in our body, we have T cells and B cells. Actually, T cells serve this important function of identifying the cancer cells and targeting them. Now, not what happens is that these cancer cells are very smart. They have learned to outgrow, learn to live even in the presence of T cells by different mechanisms. Now, either they would evade the recognition by the T cells. There are certain proteins that the cancer cells will stop expressing on themselves. So the T cells are present in the body, but they're not able to recognize them. Or they will release certain chemicals which will suppress the T cells. So by different mechanisms, even though the T cells are present, they are not able to effectively eliminate the cancer cells. Here is where genetic engineering has advanced, and what they did was they took out the T cells and they manipulated the T cells to express antibodies. Now, antibodies are something which are they're proteins which are sticking out of the T cells, and these antibodies are very specific towards the cancer cells. So you identify the proteins which are expressed selectively on the cancer cells. You introduce these antibodies into the T cells, make the T cells to produce the antibodies. Now the T cell is not dependent. So the T cell has a very efficient mechanism to identify these cancer cells. So that's the beauty of genetic engineering. And additionally, you introduce another capability into the T cells where they are able to proliferate much more rapidly in the body. So these two features are introduced by ways of genetic engineering, and this is what is chimeric antigen receptor T cell therapy. It requires obviously specific targets. Not every thing is targetable. So if you identify good targets where the target is only selectively expressed on the cancer cells, not so much on the normal cells, and it is also expressed on all the cancer cells. So sometimes you can have a problem that only there could be a patchy expression of the proteins. So these are the instances where, there are few targets who so far been identified, and they've been also successfully been shown to work in the clinics where you target those proteins and you're able to efficiently kill those cells. So CAR-T cell therapy like that has succeeded in against CD19, which is a protein and which is expressed by B cell malignancies. There's another protein called BCMA, which is expressed by multiple myeloma cancer cells. And there are a lot of other proteins which are being tested there in the pipeline. So this is how CAR-T cell therapy works. Now, from where this idea came, this has been a work in progress for several decades, and it's not only. It's a global problem. People around the world have been trying to manipulate the immune system in order to make it more functional and target against the cancer cells. It has been going on with the potential role of cancer vaccines. Then people worked on manipulating the T cells. In fact, the earliest reports were way back in 1990s. There was a group in Israel who was working on this. So there was limited success earlier on. But as the genetic engineering advanced, we started seeing some promise in early 2000s. And in fact, the first patients were dosed into late 2000s. The clinical trials started in US, and the first approval for this therapy came in 2017. In India, the scientist whom we work with, Dr. Rahul Purwar, he's an expert in T cell biology as well. So his research, right from the PhD days, focused on T cell biology. So he was interested in developing a CAR-T cell therapy. So he started his work in 2013 onwards when he returned back to India. And that is when in 2015, we joined with them as TMH, collaborated with them. And that is how we got onto this whole journey of developing CAR-T cell therapy here.
How awesome is that? And we sitting in such a fantastic point of time where we are able to manipulate our cells and even augment them, giving them these extra powers to, kind of fight against these diseases. Will be great if you kind of, explain the process of engineering these T cells for, CAR-T therapy. What are the key challenges in the processes, and how is ImmunoAct? You mentioned about Rahul Parwar, and TIFR and IIT Bombay addressing them.
Yeah. So in simple terms, what needs to be done is you have to isolate the T cells from a patient. So the process is actually similar to a blood donations, where you collect a certain amount of blood product from the patient. It's typically about 100 to 150 ml of blood product. Then what you need to do is you need to then separate out the T cells. Now there are there's different ways of doing this. There are some magnetic beads which are available. What one does is that they introduce it into the product, and these magnetic beads will specifically bind to the T cells, and then the remaining cells can be washed off. Now, once the T cells are selected, then what you need to do is you need to then introduce the gene of interest into the T cell. So this insertion, the gene cannot be simply introduced. You need a vehicle to carry it. And these vehicles are typically viral vector, because viruses in the nature are the best form of best transport vehicles for inserting the genes into the cells, because they itself have a very small genetic material, and when you introduce it into the cell, they go into the nucleus, and the genetic material gets integrated into the host cell. So in that sense, lentiviruses, retroviruses have been identified. So they have been. So you take replication incompetent lentivirus. In the sense, you don't want lentivirus having a mind of its own and also start to multiplying and causing problem. So you use replication incompetent viruses. You load them with the genetic material of interest. Of course, the design of the genetic material is another complex technology of its own. But once you've designed the genetic material and the gene construct, you load it onto the vector, and then the vector is introduced into the T cell, and then you allow the vector to do its work. So to get this genetic material into the nucleus of the cells, and then once that happens, then it will start expressing. So given the conditions, it requires certain conditions for the cells to grow, multiply. So it will happen. Now, this typically happens over a period of 7 to 14 days, this whole process that I'm describing you. But the development of the gene construct, development of the processes, standardization, that takes several years. In fact, it took Dr. Rahul's team almost close to a decade for them to go through the whole process. And once that happens, and once you have reached the capabilities of manipulating and causing the gene to be expressed in the form of protein onto the T cells, then you have to do this whole process in a very stringent, it's called GMP environment. So this is the highest forms of cleanliness, where in a clean room facilities, you do this manipulation again, and you again demonstrate that this is possible. And once you have done that, then that is when the clinical trials start. I mean, of course, you have to do the preclinical work. So even before that, you have to test it against the cancer cells in the lab, use animal models, and show that this is working and this is safe. And only once you've done the whole process, which almost took us a decade, then you use it in the patients.
Right. So it's gone through the entire process of clinical trials to right now being approved and being used on patients. What's the response so far? I mean, and what types of cancer is CAR-T therapy most effective against?
Yeah, so this CAR-T cell therapy is always dependent upon which antigen you're targeting. So CAR-T cell therapy is a broad platform. I mean, you can use it to manipulate and target different antigens. The one which we have worked is on CD19 as a target. CD19 as a protein is expressed on B cell malignancies, so B cell ALL, B cell lymphomas, and this is where the therapy is currently approved. On the clinical trials, when we treated these patients, we had an initial response in almost 70% of the patients. So these were patients who had relapsed disease, who had refractory disease, meaning they had failed their standard therapies. And the results with the current available alternate options, is very dismal. So just to give you a sense, less than 20% of these patients would even have an initial response to these available standard therapies. So from 20% to go up to 70% was a major jump for us, even in terms of initial responses. Of course, like in cancer, you don't say cure unless you've had this response going on for at least 2 years, because cancer cells have a tendency to come back, because sometimes you would just see a temporary response. There will be some cancer cells still lurking in the body, and then they would come back, eventually leading to a full-blown relapse. So that's something which we are very carefully monitoring. But initial responses were 70%. We are seeing in leukemia cohort, the responses are holding in about 50 to 60% of the patients, even after 12 months of follow-up. In the lymphoma, the number is around 25 to 30%. But still, these are quite an improvement for us in terms of what is available. Things need to be done to further improve the outcomes, but we have seen a significant jump in terms of where we were earlier.
So you mentioned in the course of the conversation that you've been using these viral vector for, the delivery of cells into the nucleus. What are the potential concerns over there, and are there any side effects of CAR-T therapy, and how are you managing them?
In theory, one of the biggest concern, as I said, is that we have taken replication incompetent viruses, which means that the job of the virus is only to introduce the vector to the gene into the cell, and then not start multiplying on its own. Because if it starts multiplying on its own, then it can cause illnesses, right? So that's why this is very carefully selected. And replication incompetent viruses, they are tested in the lab for a long period of time, and also in vivo. Once we have given the cells, also we keep monitoring them. For all the trial patients, we monitored them for these activity of these viruses, and we showed that they were not competent enough to multiply on their own. So that's one. But having said that, this is something which we keep on following the patients for. I mean, we have to keep on following these patients in the long run to see whether they don't develop second problems because of the introduction of viral vector. But the use of lentiviral vector has been around for several years now, and it has been shown to be safe in that sense. That's one. But when we talk about overall, what are the side effects of CAR-T cell therapy? There are a few side effects which are which one has to be aware of. So because the CAR-T cells they are programmed in the sense that they will multiply much more vigorously, and that's how they're going to work as well in terms of killing the cancer cells. So what we have noticed, and what is known, is that they can lead to something called a cytokine release syndrome. So when the T cells proliferate much more very rapidly, it will release certain chemicals called cytokines into the circulation. These chemicals can cause fever, they can cause drop in blood pressure, they can cause drop in saturation. So the patient can become breathless. What we have seen is with this advancement in the technology and the one which we have worked with, this incidence of this CRS is actually, the severe CRS is quite low. So you still say mild CRS where you could just have fever, you can have a little bit of breathlessness, a little bit of drop in blood pressure, but this is the severe forms of CRS where the patient requires intensive care stay, etc. Those are less. Also with improvement in patient selection. Now, if you wait for the CAR-T cell therapy to be chosen as the last option after exhausting everything, then again it really doesn't work so well. But if you're selecting them fast sooner, now with experience, this is what happens. Once you get a hang of how this therapy works, this is true with any cancer therapy. You tend to use them earlier, and then again with your patient selection, patients are in a better general condition. Again, the toxicities are much less. But cytokine release syndrome is one of the bigger concerns. The second concern would be infections. This could be there for a relatively longer period of time, particularly mild respiratory infections or sinus infections. So that's something which one needs to be aware of. Cytopenias, like the blood counts can go down and they can remain low for a while. And this again is another concern which is globally recognized and something which people are focusing on. What is relatively rare but very important, and also something which has caught the attention of the media worldwide, is the risk of second malignancies. Now, CAR-T cell therapy, like any cancer therapy, can cause second malignancy, and this is not a function just of CAR-T cell therapy. It's also because of all the therapies that were given to the patient previously. Now, typically, these patients whom we see in the clinics would have at least received 2 or 3 rounds of chemotherapy, radiation, etc. So they are already predisposed to develop second cancer. And then what happens is that when you give CAR-T cell therapy, again, this risk is there. The patients could manifest after that. So it's very difficult right now to attribute whether this is purely because of CAR-T cell therapy or because of the preceding therapies. In terms of the incidence, whatever we have seen globally from the bigger data sets, it is about 2 to 3%. This is what is expected in any cancer population. So that. But this is again something which we are monitoring, and as we increase our follow-up and if we keep on following up patients, we will also get to know what exactly the situation is.
Right. Can you update us on the ongoing research, and are there any plans to kind of expand this cell therapies to solid tumors?
Yes. So initially the research, so I mean, once we have, so I think the research can be divided into 2 parts right now. What we are doing with CD19 itself is a big research right now that we are undertaking, and then we are looking at other targets. So in terms of CD19, as I said, the first thing and foremost thing for us would be to move this therapy earlier. Like, so some of the patients who were treated on the clinical trials have even failed 6 lines of therapy. This is so these patients typically, when you fail, you're exhausted, your cell quality is not good, because ultimately CAR-T cell therapy depends upon what kind of T cells you get from the patient. Now, if you're heavily pretreated with chemotherapy and radiation, your T cells are exhausted, and therefore the product also may not be great. So we are moving this to earlier lines of therapy. We have written up a protocol, and the proposal is under review in acute lymphoblastic leukemia, where we believe that giving it in second-line therapy would work, would make it much more efficient. It will be safer for the patient, and it will also save them from getting repeated rounds of chemotherapy. So that's one we are working on. We are working on similar things in lymphoma as well, where we are trying to combine it with other immunotherapy and see whether that can improve the activity. So these are all the academic trials under progress, and we are working on them as a multi-center collaboration with other centers as well. The other targets that we are working on, the most promising target that we have is BCMA, which is for multiple myeloma. We have finished our preclinical work. We have put in an application to the regulators in Delhi, the CDSCO, and that is up for consideration. And we were waiting to hear back from them. Once that happens, we hope by this end of this year, we will start the phase 1 trials in myeloma as well. There are other targets, like you rightly pointed, solid tumors is one of the biggest concerns. We are trying to work on solid tumors as well. So there are 1 or 2 potential targets that we are working on. And lastly, we are also hoping to start our work soon on autoimmune disorders, because refractory autoimmune disorders is again another challenge, and that's a significant burden to the patient and the family in terms of both morbidity mainly. So that is again something where we are looking towards working at. Yeah. So these are all the things which are in the pipeline.
More power to you, Doctor, and the entire team at ImmunoAct, IIT Bombay, as well as TIFR. Can you share any success stories or significant outcomes from the clinical trials of ImmunoAct's CAR-T therapy?
Just to correct that, this is TMH, it's not TIFR. So there are 2 different organizations.
Okay.
So Tata Memorial Centre is the cancer hospital. In any case, so there have been several success stories. See, at the end of the day, this is a very difficult population to treat. So there are going to be a lot of there are going to be a lot of heartbreaks. There'll be a few positive stories, but ultimately, this is what we strive for. We are looking for those patients who will respond well, and then we try to see ways of how we can increase more of this. But just from the clinical trial and some of the patients whom we have treated outside the clinical trial, we have had quite a few patients who've had dramatic responses who are otherwise mostly advised for best supportive care or palliative care, where we've been told that you just have a few months to live. But then some of them got on the clinical trial and have had a good response as well. So we've had, for example, a young dentist who is a mother of a child, a very very young patient, and she got the CAR T cell therapy more than a year ago now. She's in remission. She's doing well. She's gone back to her work. So that we've had a lot of professionals. So we have a microbiology technician who's doing well, who had two relapses already. So this is a third relapse, the third line of therapy, and he's also doing well. He's completed a year of post CAR T cell therapy. We have quite a few young people. In fact, our longest survivor, who has crossed 2 years now, who was part of the first trial, he's crossed 2 years. He recently fathered a baby. He's doing well. He's not, fortunately, he's not had any sort of complication, not requiring any medications. He's completely off medications. So that we have a cyclist who recently got the therapy as a part of the outside the clinical trial once the approval came. So he's a cyclist now. He's hoping to compete at the national and international level, and he's back to his activity. So that's another good thing about CAR T cell therapy. Like, it's a one and done infusion. There will be a few patients who will have infections and those complications, but there is a significant number of patients who generally respond well, and then they are off therapy, and they're able to resume their activity. He's just third month or fourth month post therapy, and is actually actively participating in trials and cycling for several kilometers a day. So those are the success stories. I mean, there are quite a few like these. But yeah, so this is something which keeps us going.
Awesome. Awesome. Really, really cool. So how does the Indian developed CAR T therapy compare to the treatment available globally? Because, I mean, at least in the press, it's there that, the cost comparison is one of the highlights. What are the other highlights which kind of makes the CAR T therapy
I think,
developed?
yeah, I think that's a good question. It's from the scientific point of view, which is very important. That the initial therapies which were being developed are murine therapies, and even many of the people globally also have now started looking at humanized CAR T cell therapies. We fortunately already had this. This awareness that there's murine CAR T cell therapy which is available, but then we also knew that murine CAR T cell therapies were little difficult to handle, and in terms of high risk of complications and complications being much more serious. So it would have been difficult for us to have those kind of therapies in our setting, because, see, in our setting, we always have an issue of logistics. Anything which is going to add to the cost, anything which is going to add to the stay in the hospital or to the mobility will make it a non-starter, will make it very difficult for us to deliver, because our systems are always overburdened. So while even doing the research itself, Dr. Holstein, credit to them that they identified that the humanized CAR T cell in the lab was actually showing lesser toxicities. So therefore, that is what was taken forward, and now there are other groups also, both in China as well as in US, who have shown that humanized CAR T cell therapies are much more safer. So it is, in that sense, from what is already approved in the West, this is a humanized CAR T cell therapy. Therefore, it has a much better safety profile. In terms of efficacy, this is similar to some of the products which are available. At the end of the day, CD19 CAR T cell therapy, though these are all CD19 CAR T cell therapy, they will all have these small differences, and all these small differences can make them more effective, can make them slightly less effective. So a lot of variables which go in, but broadly speaking, this seems to be working as well as the other therapies which are available. But in terms of safety profile, this is much better. So it makes it easier for us to handle those therapies in the clinics.
Right. If there are patients or family who want to kind of avail this facility, where do they reach out?
There are several centers, fortunately, right now in the country who are offering CAR T cell therapy. I think the best would be to discuss with the primary oncologist or hematologist, whoever is treating them. And there's a possibility that they may already have this, or they may be already in talks and setting up their own program. We at Tata Memorial Centre are always happy to see patients given second opinion, and also direct them to any local center which should have this. The setup already in place and the feasibility of delivering CAR T cell therapy. So they can even reach out to us as well.
So what role do you see, gene technologies like CRISPR, Cas9 impacting the development of next generation cell therapies?
So this is all exciting. This is an exciting period in the cell and gene therapy space. There are a lot of things which are being done in terms of improving the scalability, in terms of improving the output, in terms of making it more easy, more user-friendly, making it more safe as well. So one of the things would be gene editing technologies. In theory, yes, these technologies could make it much easier for us to develop the CAR T cell therapies. And also not only CAR T cell therapy, for other gene therapies as well. The promise or the most attractive part of gene editing technologies is that, unlike a lentiviral vector where you have to introduce something into the cell, you don't really have to do that, and you could be more precise in terms of where you're doing the editing. But that is in theory. In practice, it may not necessarily translate into that. But the good thing is that we have an access to these therapies, and of course, as we go along, there'll be many more other similar technologies which will help us in making this whole process much easier, so that we would not take 10 years to develop one therapy. It becomes much easier for us to develop therapies, and then we are able to quickly test multiple targets and do research at a much faster pace in different spaces for cancer, be it for autoimmune disorders, etc. So I think in that sense, yes, this looks very promising. This is an exciting time in cell and gene therapy field.
Doctor, what's the role of AI you seeing over here? Because I mean, globally, I mean, artificial intelligence is completely disrupting, all possible industry verticals, whether it's business, whether it's entertainment, whether it's gaming, and even healthcare. So how do you see artificial intelligence, being harnessed to accelerate the development of cell therapy?
So one of the, I'll say more from the clinical side. I am sure that even from the cell and gene therapy side or the development side itself, this will make a big impact. But even from the clinical side, one of the most important places where we feel that AI might help is in terms of selection of patients. So currently, our models of prediction of responses are very are based upon a certain number of variables. You look at certain clinical aspects. A lot of it is actually your gut feeling. A lot of it is that is how I would put it. So a lot of it is based upon certain parameters which are not always highly accurate. So what AI can do is can make it more accurate. So your prediction could become more accurate. So you may be able to predict which patient is likely to benefit with a particular form of CAR T cell therapy. This again has been in theory. This has been one of the most promising futuristic value of AI in terms of whole of medicine, that you could tailor therapies according to the patient's profile, and you could take a lot of parameters into the consideration. So it doesn't necessarily have to be like a few variables which a human mind can process. You could take hundreds of variables of this patient, be it the blood reports, the scan reports, the clinical profile, etc., and then make a more accurate prediction of whether this is going to help or not. So that is one where I feel that at a clinical level this could work. But again, from a manufacturing point of view, it could help in the process of development. Like a lot of this is trial and error. But if the AI can help you making accurate predictions, accurate sequence development, accurate target identification, that again could help in fast tracking the whole process of development of these therapies.
Right. Is there any advice that you would like to give young researchers interested in pursuing a career in immunotherapy?
Yeah, I would say that this is very exciting research. So I think the most important thing would be perseverance. So you have to be patient when you're doing research, and it's a process which is very important. So you have to go through the whole process. Obviously, you will not have the control on the end result. So whether therapy will succeed or not is not something which you know beforehand. So that you need to have that awareness and that humility that, okay, there is a therapy that you want to test. You will have to go through the whole process. And it may not be always very attractive and fancy like how you imagine research to be, right? So it's not like you'll just wear an apron and suddenly something is going to just come out of the lab. It's not like that. It takes a lot of patience. It requires a lot of skill sets. It requires skill set beyond science. You need to know how to collaborate with people, how to work with people, how to get the work done. You need to know how to present to the, get your grants, present to the regulators. So there are a lot of challenges there. So that's something. But this is very important, because once you finish, you go through the whole process, you realize that this is all worth it. Because when you hear the stories of, I said the success stories of the patients, those patients wouldn't have benefited if you had not gone through the whole process. And several more patients. There's something which has opened up cell and gene therapy for not only India. This is for the entire southern hemisphere, the entire LMICs. You would be surprised to know rather, that there are only few countries in the world who have an access to cell and gene therapy, and even fewer who have their own technology with them. So India now has come into the league of the countries which not only import cell and gene therapy, but can have developed their own cell and gene therapy and can use that cell and gene therapy. And there have been several countries who've approached us or ImmunoAct, Dr. Rahul's team, to get an access to these therapies. So I think all of this wouldn't have been possible if we had not gone through the whole process. Right. And as a researcher, we know that if you're going to work on 10 projects, there are going to be 1 or 2 projects which are going to be successful. But then it is worth for those one or two success stories. It is worth to take the pain of going through the entire 10 project. So I think that is the most important message for them. And sorry, the last important, most important thing is that we can make it in India. So it's not necessary that you always have to go out to US, to Europe, to China to develop these therapies and do groundbreaking research. Cutting edge research can be done. It is all around us. We just need to get people together and work on this relentlessly to make these therapies happen in India.
What makes me excited about the future is that how Indian researchers and scientists are standing up, because so far it's always been the case that, for acquiring any tech, which is deep tech, be it in the space of business or healthcare, I think we always had this where we reached out to the Western nations, but I think that entire thing is changing, and that's so really cool. Education ecosystem is changing, and it's breeding more and more. I mean, the software obviously has been like one of our plus points, but I think even the government is like supporting the hardware ecosystem, and like you mentioned, cell and gene therapy is like, instead of importing, we are building our own cell and gene therapy. So that's really really nice to hear, and I'm sure that the next few years, the entire healthcare industry is going to get upended with this personalized healthcare with AI kind of converging and so on and so forth. Would you be able to kind of give us like how you see like CAR-T therapy evolving in the next 5 to 10 years?
So I think one is that the CAR-T cell therapy is in all likelihood move up in terms of where it is positioned right now and be given much earlier. That of course will depend upon the results of the subsequent trials that we conduct, and also it will depend upon the ability to give these therapies in a large scale. So manufacturing becomes a challenge. These being personalized therapies, there's always a time which is required in the manufacturing. So your abilities to deliver and give it to many more patients is going to be a challenge. But I think that is where we are. All the processes are directed towards that you're upscaling, you're increasing your technology skill sets, so that you're able to cater to many of these patients and also further reduce the cost down. So I think that is one where it is going to go in all likelihood. Second is, with better technology, I think the safety is going to increase. And the third thing is, what we are going to do is we're going to learn on how to combine CAR-T cell therapy with other forms of, be it chemotherapy, be it immunotherapy, because in solid tumors, it's still not been a success story. And so that's where I think there's some promising work happening at Pennsylvania. It's also happening at MIT, where they have been now trying to combine it with other forms of immunotherapy, like, for example, give a cancer vaccine or combine it with other immunotherapy medications which are available. So I think that is where that is the direction in which this field is heading towards. And we are I mean, solid tumors, success in solid tumors would be something which will be a real watershed moment in the field. So I think that is where we are looking very positively towards, and we are also trying to work towards it as well.
What's your personal moonshot? What are you, like, that dream goal that you're going after? And will be great if you could paint a picture of what the future of healthcare is going to look like in the next decade.
So our goal right now as a group is to see how we can provide access to the patient. So any patient who walks in and requires cell therapy, we don't deny the access to the patients just because of lack of funds. So our goal is right now to improve the access. And a second and the most important goal is how we can position it in much earlier lines of therapy and can do away with some of the side effects and intensity of these chemotherapy drugs, which many of our patients struggle to go through with. So that is where our goal right now is, particularly in leukemias and lymphomas, and hopefully in the future in myelomas as well. Because even with the advanced technologies that we have, the relatively advanced healthcare infrastructure, and I mean, some of the brilliant minds in the healthcare space we have in the country, in terms of delivery, we are always struggling because of the sheer numbers that we have to handle. And some of the most advanced therapies and immunotherapies, we are not able to deliver or give to our patients. So, for example, less than 5% of our patients get transplants where it is needed. And so we want to reach a point where many of our patients, at least a significant number of patients who require these cell and gene therapies, will have an access to them, and we will be able to deliver to them. We also want to work with the other centers in the country because we understand that TMH has taken a leadership role here because by virtue of its early commitment to this program. So we understand that new programs will come. We ourselves have benefited by learning from experience of others. So we want to pass on this experience to other groups, and we have been working on different areas in terms of creating forums where these discussions can be done. This is happening on a regular basis now. And also to initiate collaborations where we could do multi-center trials, because smaller trials can be done in a few centers, but when we go into a larger trial, we will need multi-center collaborations. That is where our groups like the Hematology Cancer Consortium, the National Cancer Grids, which have been in place for the last few years, will help us take this forward. So this is where I feel that the healthcare is going to go. Healthcare is going to improve. We are going to get access to many of these therapies, and this is an important time for us, because historically, whenever a new therapy is developed anywhere else in the world, we have to wait for the company to get that therapy to India, and typically it takes more than a decade for at least in the hematological malignancies, because hematological malignancies is considered as a rare disorder because of the numbers, the fewer numbers when you compare with other malignancies or other disorders. So the companies generally see it's either US, it's Europe, it's China, and then it's rest of the world. So we are a part of the rest of the world. So we hope that this changes, and cell and gene therapy has given us this opportunity to stand on our own and create the therapies that would be helpful and accessible easily to our patients.
Doctor, really appreciate you taking time and being part of the podcast. This is such a groundbreaking moment for us in India. Globally, there are some of the brightest minds you mentioned, working towards, creating successful CAR-T cell therapy. And here we are in India, developed it and tested it, and it's we also democratizing it, bringing the cost down, and I hope that the conversation changes, because you mentioned in the course of the conversation that whatever cell therapies, we've always had to wait, we won't be able to wait, and I hope that we become the exporters of the world, share our expertise and our therapies so that, it's available all over the world. So really appreciate you taking time and being part of the podcast. Wish you and your team the very best. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys later. Thank you. Thank you, Doctor. Really appreciate this.
Thank you so much.